In this practical exercise, we are going to construct a phylogenetic tree to illustrate the evolutive relation among a group of globin sequences from different organisms. First, we have to select a target sequence of interest we want to investigate. In this case, we are going to the NCBA webpage and search from the protein database from the human hemoglobin sequence. Here we have a series of hits. This corresponds to Homo sapiens, but it's partial, so we go to the reference sequence and obtain the corresponding complete sequence of the hemoglobin subunit beta of Homo sapiens. So once we have our first sequence, we use the BLAST tool to search for homologous to the human hemoglobin. In this case, we are interested to compare hemoglobin sequences on a predetermined group of organisms. So, under organism, we select the species that we are interested to compare. Select human, mouse, orca, chicken, falcon, alligator, and zebrafish. Once select the organism of interest, we perform a protein blast with default parameters. Here we have our results, and we proceed to select those sequences corresponding to the proteins of interest. The hemoglobin subunit beta from Homo sapiens, the hemoglobin subunit beta from mouse. Here we have several isoforms. For the mouse, we select those that correspond to the query. Here we have the hemoglobin subunit beta from the orc. In this case, we have to select a predict sequence that is probably derived from a genome annotation. We should try to avoid the use of predict sequences because in the majority of the cases, those sequences has been not confirmed at the protein level and they come from automatic annotation process. But in some cases, if the organism has been little studied, it is the only information that we have. The hemoglobin from chicken, same from the falcon, alligator, and zebrafish. Once we have selected the homologous sequences, we proceed to download it as a FASTA file. Here we have the file with the selected sequences, and we proceed to modify the first line corresponding to each sequence to improve the visualization and posterior analysis. And so on. Here we have an alignment, a sequence file with the names modified. And now we are going to proceed with the construction of the phylogenetic analysis. For doing this, we are going to use a web server for phylogenetic reconstruction. This program runs and connects various bioinformatic programs to reconstruct phylogenetic trees from acetal sequences. It includes multiple alignment procedures, several methods for construct phylogenes, and programs for the visualizations of the trees, and additional some utilities for the manipulation of the sequences. As we comment in classes, there are many programs to build phylogenetic trees, and you can use the best who fits your needs. So, here we have three choices for the selection for construct a phylogenetic analysis. The first is a black box procedure when you are not able to customize any of the steps in the construction of the phylogeny. In the second option, you have the possibility to customize some of the parameters, and in the third option is a fully customizable phylogenetic workflow, and we are going to select this option that allows to create a workflow. We name the analysis, we select a program to construct the multiple sequence alignment. In this case, we're going to select the crustal. The second step deals with the curation of the alignment, and you can customize the regions in the alignment with poorly aligned positions, such as insertion or deletions that could have a negative effect in the quality of the produced tree. We're going to leave G-blocks as the default option. And in the third step is the selection of the method for the constructing the phylogeny. Here we have a maximum likelihood method, a parsimony method, and two distance methods. We're going to select the neighbor joining distance method. Finally, there are different tools for the visualization of the tree. We keep the first and proceed in a step-by-step way to visualize every step of the process. So we proceed to create a workflow. Here we have to introduce the sequence. Here we have our sequences, and submit for the multiple alignment using the crustal software. Here we have to wait until Crustal finish to align all the sequences. Here we have the alignment result with marks in the conserved positions among all sequences. You can download and also edit the alignment. Also, you have information about the guide tree used by Crustal to construct the alignment. This is not the phylogenetic tree. It is an indication of the relation between sequences, but not has any statistical support. You can remove sequences from this tree if you observe that some sequences are too distantly related or too closely related, and consequently could affect the final result. We are leaving all sequences and proceed to the next step. Here we have in the curation step that deals with the removal of poor aligning positions. In this case, the sequences are very similar, so we don't observe big regions with gaps. Thus, we employ a less stringent selection and submit the curation process. And wait for the curation process finish. Here we have the curation results. We have the sequences with the conserved regions color code, less conserved regions marked differently. We have some statistics about which regions the program is going to use for the alignment. In this case, the sequences are very similar, so going to maintain almost the complete alignment for the next step. It is the guide tree after the curation, and we continue to the next step. Now we are ready to launch the phylogenetic calculation. Thus, we select the parameters for the neighbor joining distance method. We can select the statistical estimation of the accuracy of the inferred tree by the use of a bootstrap method. In this case, we are not using the bootstrap method because it takes too much time. Then we can select the substitution model of the amino acid changes. We have two matrices for select. We're going to choose the Diehoff matrix and submit the phylogenetic analysis. Here we have the resulting tree of the relation between our sequences, and for a better visualization, we are going to render in a visual friendly mode. Thus, we proceed to the final step using the program 3D to allow us to customize the visualization of the resulting tree. Now we have the resulting tree that displays the evolutive relation between the different globin proteins in the different species. As you can see, the mammals' sequences are clustered together, same as birds, which also include the reptiles in the same clade, indicating the common ancestor that birds and reptiles share a common ancestor. And finally, as outlier, we find the most distantly related globin sequence from zebrafish. We could modify the resulting tree for better visualization. For example, changing the relative orientation of the nodes, here we could change to render the most here we have birds, reptiles, mammals, and the fish. We also could modify the visualization from a classic rectangular mode to a radial, and the relations are represented in a radial way. And also you can modify the time, the font, letter, and other parameters for better visualization. In this way, we have conducted a very simple phylogenetic tree that displays the evolutive relation of the globin proteins between different organisms: mammals, birds, reptiles, and fishes.